Hello, everyone. Welcome to ASGCT's Around the World, focus on technological frontiers in South American gene and cell therapy, from viral vectors to engineered cells. This event will provide an in-depth exploration into South America as an emerging and dynamic player in cell and gene therapies, set to revolutionize modern medicine on the global stage. This virtual program explores the technological frontiers shaping the development and deployment of advanced therapies across the continent. So we'll have the presentations from our speakers, and a short Q&A period will follow all the speakers' presentations. You're encouraged to type questions into the Q&A throughout the presentation using the chat tab located along the right-hand side of your web page. You may continue to submit questions throughout the whole Q&A discussions as well. Okay, so I would like to introduce today's speakers. To begin, I'm Dr. Guilherme Baldo. I am your moderator for today's event. I am an associate professor in the Department of Physiology at URGS, Brazil, and my research focuses on gene therapy for rare diseases, including inborn errors of metabolism and neuromuscular diseases. Our first speaker today is Dr. Osvaldo Podaiser. He earned his PhD, his BSc and MSc degrees at Ben-Gurion University in Israel, and obtained his PhD in biological science from the University of Buenos Aires. He pursued postdoctoral training at the Leloir Institute in Argentina and at the Institute of Genetics and Molecular and Cellular Biology in Strasbourg, France. From 1996 to 2008, he was professor of cell biology and applied genomics at the University of Buenos Aires. Since 1997, has led the Laboratory of Molecular and Cellular Therapy at the Leloir Institute. As member of the CONICET researcher career since 1992, he reached its highest rank on superior research. His research has centered on the development of oncolytic viruses for cancer therapy and application of genomics to precision medicine. Dr. Podaiser has authored more than 130 scientific publications, delivered nearly 200 national and international lectures, including several keynote presentations, and is a co-inventor of close to 20 patents. Thank you, Dr. Podaiser, for being here.
Okay, first of all, I would like to thank the organizer for the invitation. It's really a very exciting meeting, and I hope you will love what I'm going to tell you about our work with the use of oncolytic viruses for tackling cancer heterogeneity. The main drivers of our work from one side was tumor heterogeneity. This is very simple for people working in cancer. Tumors are really heterogeneous. And the most important thing is that there are cancer subpopulations of cells that are antigenic differently, and they produce different antigens. And in particular, when the metastasis appears, in most of the cases, the metastases are really different from the primary tumor in terms of the antigens the metastatic cells express. And on the other hand was the idea of personalized targeted therapies. That means that instead of using the concept of one drug fits all, it uses a specific drug that targets a specific feature of a cancer cell. And the problem with that is that those cells, subpopulations that bypasses the effect of the drug will repopulate the tumor and allow the tumor to metastasize, to evade the treatment and metastasize. And the problem with combining targeted drugs is that if you use them at the optimal dose, there can be an increase in toxicity. So using that concept, we moved with our own work with oncolytic viruses. And in the field of oncolytic viruses, you can roughly said there are two different groups. Non-specific one, such as Newcastle, uh, those viruses attack mainly cancer cells because they have a weak interferon-mediated antiviral response, which is not true for all the tumors. There are few tumors that probably, such as brain cancer, that probably have a weak an antiviral interferon-mediated response. And on the other hand, the more specific viruses that are transcriptionally targeted, and I showed that here, for instance, for adenovirus, you can see the E1A gene, which is essential for allowing adenoviral replication, in which we remove the E1A promoter and instead we incorporate a tumor-specific promoter, which in fact is being selected from genes that are actively engaged in expression in the tumors. So in this case, the problem here is, again, if you use a single tumor-specific promoter or TSP, then you will have only one type of one subpopulation of cells that will be targeted, and then only one type of tumor antigens. In case you can produce a hybrid tumor-specific promoter or TSP, then you would probably target more than one subpopulation, and there would be a release of an extended number of tumor antigens, a broader number of tumor antigens. And based on those two premises, which is tumor heterogeneity and personalized treatment, what we did was a bioinformatic analysis to try to see whether we can identify genes and from them isolate the promoters that can serve to prove our hypothesis, meaning that we can produce an hybrid TSP, and with this hybrid TSP, we can broaden the targeting of our oncolytic viruses or oncolytic virus, and then target more than one cancer cell subpopulation. And after this bioinformatic analysis, we isolated or identified two genes, VWA2 and A33, by checking messenger RNA, isolate the promoters, and checking microarrays in 11 primary tumors and then 10 metastases that, in fact, those that were obtained from human samples, that in fact A33 is mainly expressed in primary tumors and decreased in metastases, while the opposite was true for VWA2. So we combined the two in a single tumor-specific promoter. In this way, we isolate a part of the VWA2 promoter, which does include the Tata element, all those boxes, all those transcription factors that allows the RNA polymerase II to be active, and clone that upstream of the A33 promoter, in which we kept all the RNA polymerase II transcription factor sites. And then we produced three different oncolytic viruses. One of them, which is driven by the A33 promoter, and this is the name, AV22EL. The second one, whose replication was driven by the VWA2 promoter, and in this case is the whole promoter, so that virus is active as well. And a new virus, which was the AR2015, which is the one in which the replication of the virus is driven by the hybrid tumor-specific promoter that combines a fragment which is transcriptionally inactive with the ATTT promoter. And then the first was to try that in 4 different human colorectal cancer cells, in which you see that in fact AR 2015 is active in all the 4. The most important thing is that HCT cells that do not express A33, so in these particular cells, AV22EL that is active only in those cases in which the cells express A33 is not active in HCT cells, confirming that in fact the tumor-specific promoter is active only in those cells in which the mRNA can be detected. So, on the other hand, AR 2015, which is the yellow one, was active despite the fact that those cells do not express A33, and it seems that the transcriptionally silent fragment of the VWA2 promoter confer the capacity to this hybrid tumors specific promoter to allow viral replication. AR 2015 is not active in normal intestine cells, neither in fibroblasts, microendothelial cells, and neither in human melanoma cells. So what we can see here is that AR 2015 is highly specific and is inactive in all normal cells we have tested, and even in non-related human melanoma cells. Then we set up a metastatic model in which HCT cells, when injected in the spleen, cells migrate to the liver, produce metastatic foci. 15 days later, when we already knew the foci were already developing, we start treating mice, and in fact, we observe that only those mice that received AR 2015, combined or not, with mainstay chemotherapies such as 5-FU, were the only one in which the mice were still alive after 30 years, while those mice that received, the control mice or those that received 5-FU alone or AV22EL, oncolytic virus, all were removed from the experiment because they reached 2 centimeter cube. So at the end of the experiment, 8 mice out of 13 were completely cured. We weren't able to detect any metastatic foci in those mice. In a second, very similar experiment, instead of using AV22EL, which is not active in HCT cells, we use as a control AV636 oncolytic virus. And despite the fact that the mice developed some metastatic foci in the liver, their size were not too big, and in fact, the area was very small, very similar compared to what we observed with AR 2015, which in this case, again, confirming that AV636 was active as expected, while the difference was that AR 2015 was slightly more efficacy, showed more efficacy in this model, indicated that it's slightly more active than the oncolytic virus, in which the replication is driven by a single VWA2 promoter. And in the third experiment, in which again the colorectal cancer cells, I didn't mention, but all these experiences were done in nude mice, okay? those that are in immunocompromised nude mice. So again, we injected cells, in this case, lobo cells, lobo colorectal cancer cells in spleen. Again, the cells migrate to the liver, produce the metastatic foci. And the most interesting thing here is what was that oxaliplatin treated mice observed no effect, while mice treated with AR 2015 or AR 2015 plus oxaliplatin, we observe a really great inhibition of metastatic foci size. And in fact, most of the mice, 3 out of 5 and only 1 out of 5 of those treated either with AR 2015 alone or combined with oxaliplatin showed metastatic foci in the liver. But most interesting was that we reached statistical significance only with the combination of AR 2015 when compared to oxaliplatin or PBS. So the main conclusions of this work was that systemically administered AR 2015 was very effective at reaching hepatic tumor foci, metastatic tumor foci, inducing a robust anti-tumor response. And interestingly, this was further enhanced by combination with a subclinical dose of oxaliplatin. And we believe that this shows that our hypothesis that we can produce an oncolytic adenovirus whose replication is driven by tumor-specific promoter can be used to tackle cancer heterogeneity. Finally, I would like to mention that this work has been led by Eduardo Caferata, which is a senior investigator in my group, and with the financial support of an Argentinian NGO, AFULIC, a grant for our National Agency for Research, Development, and Innovation, partially by the startup Tervax, and there is a related patent application. Thank you very much.
Thank you, Dr. Podeiser. Just a quick reminder that we have our Q&A. If you have any questions for Dr. Podeiser, we have our Q&A chat where you can type your questions. Our next speaker is Professor Maroon Khoury, with a PhD from the University of Montpellier and postdoctoral work at MIT and SMART Singapore. Dr. Maroon Khoury bridges the gap between scientific discovery and patient care. He's a professor and principal investigator at Universidad de los Andes in Chile and serves as a chief investigator office of Cells for Cells, a university spin-off advancing cell-based therapies. Dr. Khoury has contributed to more than 10 clinical trials exploring regenerative treatments for conditions such as osteoarthritis and heart failure. Since 2021, he has led the Impact Center for Precision Interventional Medicine, where teams are combining artificial intelligence, tissue engineering, and next-generation cell and gene therapies. In 2024, he also co-founded Evast Bio, a clinical stage biotechnology company pioneering the development of small extracellular vesicle therapeutics. His current work centers on developing living drugs, including mitochondrial transfer therapies for blood disorders, such as graft versus host disease and osteoarthritis. Innovations aimed at turning scientific insights into tangible healing. Thank you, Professor Khoury.
Hi, everyone. It's good to be here. We'll be talking about the technological advances in advanced therapy, cell and gene therapy in our region in South America. Was invited by the American Society or for Gene and Cell Therapy and IBT Cell in their annual meeting. So seeing the two society collaborating is really interesting, and I'm really happy to be able to give you a regional case study building a cell and gene therapy hub in Chile. This is my disclosure. I do have a academic affiliation, but as well, I'm the CSO of a spinoff from the same university and consortium, as well as a startup called Evas Bio that is located in the United States. So let's situate where we are in this important market cap. We're looking at Nvidia with 4.6 market cap versus all the pharma together, almost matching this amount. And then if you see the small bar here, this is where we are as cell and gene therapy worldwide, not only South America. We're at 15 billion US dollars as a market cap. So we're a tiny, minuscule market share worldwide. However, the impact is definitely important. And when we're looking at the number of products in cell and gene therapy approved in the past 2 years, this is from 2024, we can add up about 4 to 5 additional product being approved, not only by FDA, but worldwide, all countries included. We're closing to 110, 112, with mostly non-genetically modified cell therapy accepted, approved, 67%, accounting for 16 gene therapy and 17 modified cell therapy, mostly CAR T-like treatments. However, when we look at the geographical distribution, only few are approved in South America. And then there are technologies and therapies that been approved outside and then approved in versus organic innovation from the region. And why this is relevant, because when looking, for example, at the CAR T treatment and looking at which countries are treating and what are the number of patients treated per country, we can see that South America is really trailing behind all these numbers. United States leading it, followed by EU, China, and other country as well. And then the access to these treatments. We're not only talking about CAR T, we're talking about all the those expensive FDA approved advanced therapy reaching up to 2 to 3 to millions of dollars have limited geographical access. And I know even within the US, looking at the different region as well, that there are discrepancy in how patients are able to access these expensive treatments. So focusing on South America, actually Central and South America's focus, because I'm including as well Mexico, we can have a regional landscape overview by country, and I divide them into 3 categories: advanced ecosystem, developing infrastructure, emerging market, and of course nascent market, where Brazil is leading in terms of approval, in terms of regulatory framework for these treatment, and as well in terms of funding, followed by Mexico, Argentina, and Chile, which I think they're at a developing infrastructure status, where they are building the ecosystem, building the regulatory frame for giving access to these treatments. So this is a regional mapping of cell and gene therapy landscape in Latin America. Brazil, which already has many approved, commercially approved therapies, followed by Mexico and the rest. So when we're talking about cell and gene therapy, we're talking basically mostly about CAR T therapy being not only in the clinical trial, but as well for approval. This is account for most of the focus areas, including hematological cancer and lymphoma, followed by gene therapy, rare genetic disease and inherited disorder, stem cell, regenerative, orthopedic, and other treatment. This is kind of the distribution. The key player and partners in Brazil, there are important development, as I mentioned before, from all these institutions: Butantan, the Inca, the University of Sao Paulo, but as well in other countries, Buenos Aires and Colombia, and of course Chile, that will keep separately, where I will be focusing on what are the advances in Chile, where I'm residing. So what's important is to understand that the number of active clinical trials are increasing. 2023, we're accounting about 34 trials, and today we're reaching about 65 trials at least registered in the different platform. In terms of investment, we are very far from the investment done in the United States alone. So we're talking about total investment to date about 450 for all the different countries, but an important growth rate. So if we're looking specifically at the Chilean and the Latin American market in terms of CAR-T, we are able to think that this is a total addressable Chilean market is about um 1,700 patient versus 13,000 patient per year of candidate that can have access to this treatment. So it's very important to look at the strategic framework for investor engagements. So definitely there is an interest of building value in the region for cell and gene therapy market. There is a market potential, especially in opportunity in rare diseases or very rare diseases because of the population size, patient data and regulatory milestone, market growth projections. The tangible progress are, as I showed in the clinical trial pipeline and the results, we have an important growth in number in the past at least 3 years. The academic partners are very present. I will mention about public and private and academic ventures, manufacturing capacity, specifically now in Brazil, but then Colombia and Chile are following behind. Strategic advantage, as I mentioned, local expertise, cost efficiency, how much we believe it costs 60% less to do a preclinical and clinical pipeline in the region versus, for example, US or Europe. There is an interesting genetic diversity inside, and of course, an important regional health network. Funding plan could be capital, could be as well private and private, public and private, and government funds. Beyond the capital, the important value of expanding the cell and gene therapy in the region is mentorship and guidance, strategic network, and of course, bringing these treatment to improve the healthcare in the country. So, as a summary, SWOT analysis shows strengths in terms of growing interest and investment, emerging biotech hub in countries like Brazil, Mexico, Argentina, and Chile, important opportunities, including potential for regional harmonization of regulation, the reliance and our regulatory agency talk to other agencies in the region, government incentive. Now, shifting to the weakness, we feel there is a fragment and inconsistent regulatory framework between countries. There is high development and treatment costs, limit access, and the threat are economic instability affecting R&D system, dependence on imported raw material and technology. We do not manufacture cell media or reagents required for these advanced treatment, and of course, the threat is always the political as well instability, which actually govern the policy of grant in R&D. Future outlook is opportunities, expansion of the local manufacturing capability, increased access through price negotiation, regional clinical network trial network development. There are space to develop more accessible, faster to reach patient clinical trials, public and private partnership, and regulatory harmonization initiative. So these are the projection, and as a case study, I will be mentioning the work done in the initiative of Impact. Impact is a center of excellence funded by ANID, which is equal to the NIH in the United States. It's a matching fund. This means you have to bring money as well from the private sector on the table, and you have to have not only in economic impact, but as well a social impact and an educational impact as well. So we are a group of 13 PI leading different area, including cell and gene therapy, biomarkers, artificial intelligence for image processing and acquisition. And then we are trying to build that ecosystem. We're a 5 university. The main campus is University of Los Andes, sitting on the foot of the University of the Andes Mountains. And we have within our campus a university hospital, a faculty of medicine, and of course our laboratory. And what we're trying to do is different levels of innovation. And what's related to car, we're trying to bring that technology, that know-how to the country, which definitely has a lower innovation indicator, but it has a major impact on health system. While doing this, we are as well moving up in the innovation values through not only doing incremental innovation, but as well trying to reach frontier and hopefully global disruptor innovation in our pipeline. So our entrepreneurial ecosystem is public-private funding, where we're working with R&D project in a venture studio-like status. And by this I mean we have an incubator, we have an accelerator, we have GMP facility, we have a way of validation in our clinical campus, and other collaborators that are interested in using our technology. And finally, branching towards the spin-off and the startup from the same entrepreneur ecosystem for the commercialization. I have to admit, it was not an easy walk. It was from, as the media will say, from magical to mystery to here comes the sun. So this means a lot of pitfalls, a lot of challenges to overcome. And then we've been able in the past decade to conclude more than 10 clinical trials. Two of them are extracellular vesicle related. Most of them are mesenchymal stem cells for allogeneic use, isolated from umbilical cord, in different applications, including osteoarthritis, heart failure, cartilage lesion, autoimmune diseases, totaling for more than 1,200 patients. And what's the most important number is the zero serious adverse effect. First patient was using allogeneic mesenchymal stem cell was dosed in 2015, where we tried to brand it as a cell stem. And first completed and published trial was done in 2017 for the validation of these umbilical cord MSC into patient with heart failure. And this is where we're seeing a positive effect from the infusion of these cells into the patient, looking at the ejection fraction, which was published, as I mentioned, in 2017 in Circulation Research. And we've been able to, put the photo of our campus with the hard mosaic showing our data. This was an interesting flexing point for us, because it was taken by foreign media and reported in more than 14 different languages through Europe, Asia, and North America, where it was highlighted the enabling adoption and affordability of the way we're doing, because we did only intravenous injection versus other trials where they did more invasive route of injection. So this is an interesting evolution from complex to straightforward, and it was a bit of, inspiring for us, as we took it as a motto for us to go ahead and continue. So the second trial we did is using the same cell for a local route. It was intra-articular injection for knee osteoarthritis with positive result. And then we showed that this is reproducible and we can transfer it to a third party. And then this is what we did with a group in Colombia, where they were able to show that this is reproducible and it can be transferred outside the Chilean borders. Definitely, cell and gene therapy have important limitation, and we feel that MSC has there's a lot of lesson learned from the MSC experience worldwide, not only in Chile, and they build the blueprint for the next generation that could be cell-free treatment like extracellular vesicle. It's like inheriting the family talent without inheriting the family debt, and this is exactly what we pursued. Now we left the cell in the lab, made them produce extracellular vesicle, has to develop a pipeline, including preclinical testing, where we looked at safety of these EV, but as well measurable joint regeneration and protective effect, moving to a process development, being able to produce clinical grade at a small scale of these EV, showing that they conserve their identity, and finally, a first in human knee joint therapy while looking at the mechanism of action. We'll be able to show for the first time, the first patient dosed with exosome producing therapy for osteoarthritis. This is the patient. This is we're looking at the different index of pain, stiffness, and physical function before and 6 months after. The lowest the score is, the better the patient feels, with MRI image suggesting therapy potential to all disease progression. And now we're have science is based not on anecdote but on evidence, and this is why we have now an ongoing clinical trial looking at 3 different doses of these EV into OI patients. What's important is when we're talking about cell and gene therapy being accessible, it's not only wishful thinking, it's a reality for us. And for this, we created within the Center Impact a unit which is called CRIO, for Clinical Research Impact Organization, in 2024. And their job is to help clinician, but as well investigator like us, to bring their treatment to the patient. And then this is a CRO, academic CRO, I would say, that is committed to accelerate clinical research and to bring innovative therapy to the market. And today they are conducting more than 23 ongoing clinical trials. It's a small, relatively small team, but they're working not only in osteoarthritis, but as well in mental health, autoimmune disease, and cancer. And an example of things we're doing, taking in consideration that the work we've done with GVHD, the clinical case we've treated pediatric patients suffering with graft-versus-host disease, non-responding to conventional treatment, and as well the FDA approval recently in 2024 December of a product based on MSC for the treatment of these patients. We had a compassionate case of a 65 years old patient who did not respond to any line of the treatment, including GNK inhibitors. And as a compassionate case, we've been able to dose him with these MSCs. It was a fraction of the commercial price that these treatments are sold in the United States. Again, this is umbilical cord, so we do have as well proprietary patent on this one. And then he showed important improvement in fatigue at 6 months, and today at almost a year post treatment. So we believe there is a interesting momentum where the region, the South and Central and South America, is ready for cell and gene therapy innovation and investment, looking at the scientific interest, the regulatory investment, the patient access, and the future outlook. So I encourage early investor that they are able to secure a first mover advantage in rapidly expanding market, and hopefully we'll be able to see treatment like CAR-T in the rest of the region beyond only Brazil and another country. And then as the region is rich and known for its biodiversity, we hope that it will be as well known for its advanced cell and gene therapy. Thank you very much. Muchas gracias. Obrigado.
Great. Thank you, Dr. Curry. Once again, if you have any questions, please type in the Q&A chat, and we will have a Q&A live session after the last presentation. Our last presenter today is Dr. Hilda Putter Silva. She holds a degree in biological sciences, medical modality, and a PhD in biophysics from the Federal University of Rio de Janeiro, UFRJ, in addition to having completed postdoctoral studies at the University of Florida, at the ophthalmology department with William Halfworth. She was a pioneer in creating the first laboratory in Brazil producing AAV vectors, adeno-associated vectors. Her research focuses on gene therapy for neurodegenerative diseases, particularly glaucoma, using AAV vectors and CRISPR-Cas9 technologies. As a member of RENERA, she supports the Brazilian National Health Surveillance Agency, which is Anvisa, which is Brazilian FDA, in the evaluation of gene therapy products. Currently, she's an associate professor at the Carlos Chagas Filho Institute of Biophysics at UFRJ and head of the Gene Therapy and Viral Vectors Laboratory.
Everyone, it's a pleasure to be part of this ASGCT webinar. Today I'll talk about AAV gene therapy in Brazil. I will present how it has developed in terms of clinical applications, and also present the research perspective on what we have been doing in the lab. To introduce AAV gene therapy, it's important to bring the beginning of it. And it always started with those two publications from University of Florida. I would like to say that I have a great appreciation for University of Florida. In the first paper, AAV genome was cloned into a plasmid, and a virus was produced from it. And on the second one, published in 1984, AAV was used as a cloning vector, transducing mammalian cells as a vector itself. It means that it took a 30-year journey from the development of the AAV vector to the approval of the first AAV-based drug that happened in 2012 in European Union. The drug was called Libera, and it was followed by Luxturna in 2017 in the US. The ideal for any viral vector is that all or many viral sequences as possible can be removed from the original virus, and then making room for the insertion of the transgene or other therapeutic genetic material in its regulatory sequence. An example of another therapeutic material could be a siRNA and its regulatory sequence besides a transgene. And in the case of AAV, the first vector was, since the beginning, a gutless vector without any other sequence besides the ITR. And the ITR are important to encapsulate the vector sequence into the viral capsid. One of the advantages of AAV vector is the amount of different wild type serotypes represented here by the different numbers. Each serotype have a difference in the capsid protein. It means that in general, they ligate in different receptors, and because of that, it can reach different organs. And here you can see that through the different colors and reaching different organs. And the knowledge of the different capsid open up the possibility to develop new AAV serotypes artificially through different technologies, through maybe improved transduction. Another example is to overcome immune system. Here in this slide, one of the techniques is to produce a hybrid capsid is to mix the wild type serotypes, and you can see here through the different colors of this hybrid capsid. Another technology used to produce new capsid is through directed evolution. And it's important to mention that new capsids are a big trend for biotech companies right now, and also for some labs. They are all looking for some specificity for different cell types or maybe different organs. And it took some time for gene therapy to start in Brazil. Here in this timeline, I'm going to show you some important events. In 2002, some Brazilian patients were recruited to participate in a US clinical trial. It was an AAV-based medication with factor 9. It was for hemophilia. In 2020, Luxturna and Zosesma were approved by our regulatory agency called Anvisa. And only in 2001, Anvisa published the first regulatory frameworks. The first one regulated the registration of advanced therapy products and established criteria for demonstration of long-term efficacy, effectiveness, and safety. Another one regulated the conduction of clinical trial of this type of products in Brazil. 2 years later, Anvisa published another regulatory framework that established good manufacturing practice for genes and cell therapy products. And last year, in 2004, there was an update of those 3 regulatory frameworks based on updates that were done in FDA and EMA for this type of gene therapy and cell therapy products. Also last year, 3 other products based on AAV were also approved for use in patients in Brazil. I will include the timeline of my lab in here. And in 2002 was the first time I went to University of Florida to learn about AAV production and gene therapy. I returned to Brazil after a postdoc. It was in 2009. In 2010, I got a tenure position at the Federal University of Rio de Janeiro. And since then, I've been establishing a V vector core in my lab, and I've been also working on a neuroprotective gene therapy for glaucoma. And I'll talk a little bit about both. The first step for any viral vector production is the plasmid prep. We do in the lab all the cloning, we exchange transgene, we are able to exchange promoters, to do point mutations, even to do some mutations with the helper plasmid. We produce all the plasmid with commercial kits and gigapreps, and they are all endotoxin free. We use HEC 293 cells as our packing cell line. We use both 2 plasmid or 3 plasmid production system. We co-transfect the cells with PEI. And for the upstream process, we use adherent cells. We work with cell factories, 2 layers, 4 or 10 layers, depending on the amount of vector you want to produce. After 72 hours of the transfection, we detach the cells. For some serotypes, we also take the vectors from the media with PEG. After that, we lysate the cells through freezing and thaw process, and we treat this lysate with benzonase to digest all the nucleotide sequence. The clarified lysate we run through iodixanol gradient and ultracentrifugation. After that, we collect the band that is referred to the AAV and process that through a FPLC. We use different columns depending of the serotypes. They are all commercial columns, prepackaged columns. And in the end, we take out the salt by a simple dialysis process. Our vector, we do a couple of analyses of this final product. We test for endotoxin. We analyze contamination through silver stained gels, as you can see in this picture here. We quantify vector genome particles with qPCR. We are moving to digital PCR right now. And we also we do electron microscopy analysis to quantify empty full. It's important to mention we don't sell vector. We produce vectors under collaboration with different labs in Brazil. It's expensive for this production because most of the supplies come from the US or Europe. And it's also important to mention that most of these supplies take a long time to arrive in Brazil. For example, the last cell factory I bought, it took more than 6 months to arrive. It means that we have to plan in advance to be able to keep this vector core running. And now I'll talk a little bit about my research that we've been working in the lab. I work with glaucoma model for more than 10 years right now. Glaucoma is a multifactorial disease. One of the main symptoms is the loss of the vision that happens through the progression of the disease. And in this graphic, I can show here the different types of glaucoma. I'm not going into details of those types of glaucoma, but the point is to show you that besides having different types, the only common point between all of them is the degeneration of the retina ganglion cells. Once these RGC cells degenerate, the patient loses their vision, and it's independent of the type of the glaucoma. And if we can stop or delay this degeneration somehow, we could improve the patient quality of life. And we approach this through gene therapy. The transgene we have been working for a long time, it's called MAX. It is a transcription factor. I'm also not going into details about it, but it's a member of a MIC-MAX-MED family of transcription factor. And I think the important point here to understand is that MAX is a master regulator, meaning that it regulates different process in the cells. And those two results present here are very important. The retina ganglion cells have max expression when in a normal situation, and after a degenerative process, the amount of MAX decrease. You can see that through this immunohistochemistry, that 30 days after a degenerative insult, here it was increased intraocular pressure, there is a decrease of the amount of the expression of this protein. And also this analysis of messenger RNA through RT-PCR. And it means that every insult that promotes the generation of RGCs, there is a decrease of the amount of MAX. And our hypothesis was, if we can increase MAX expression, can we rescue retina ganglion cells? And because of that, I went for the first time a long time ago to the University of Florida to produce AAVs to overexpress MAX. We use GFP as a control. And we can see through this heat map here that overexpression of GFP was not able to rescue the ganglion cells after the degenerative insult. That was the high intraocular pressure. 14 days after the insult, we can see a lot of degeneration. This degenerative process is progressive through the time. However, if we overexpress MAX, we can see we can rescue those cells. The cells are not just there, but they are also functioning. And you can see this through this electrophysiology analysis in which the retinas treated with AAV MAX, they are working as the naive retinas. However, if we analyze this therapeutic effect 4 weeks, not just 2 weeks after the insult, we lost the effect. And we think that one of the reasons of this loss is because we don't have the entire retina transduced. We just have between 1/4 or 2/4 of the retina transduced. And our hypothesis here was maybe if we can improve the vector, maybe we can rescue the therapeutic effect again. And then we construct a different vector. We use a mutant of the serotype 2 that was published in this publication up here. And also we exchange the promoter. We exchange the CAG promoter to a PLE promoter that is a specific promoter for RGCs, and it also promotes a very strong expression of the transgene, as you can see here through this GFP injection and fundoscopy analysis 1 month after the injection. And then we analyze this new construction with this new promoter and this new capsid. And you can see here that we are able to keep the cells alive and functioning 30 days after the insult and also 90 days after the insult. Here we have a quantification of the ganglion cells. This blue is the vector with the PLE53 promoter, and the light blue is the other PLE promoter that both are specific and stronger for ganglion cells. And we can see we have more cells, and these cells are also working and analyzing the electrophysiology of these cells. And I guess the take-home message of this research part is that we can always improve our therapeutic effect, looking for more effective capsid or changing the regulatory sequence. We don't have to give up our gene therapy after a negative result. And to finish, I would like to acknowledge some of the professors that have been helping and working together with my group. And I would like to give a special thank you to Bill Hauswirth at the University of Florida, in the place where I learned everything I know about AAV production and AAV gene therapy. I also would like to thank the Brazilian funding agents and my students that right now are the ones really running the lab. And I would like to finish saying we have AAV gene therapy made in Brazil. Thank you to all, and I'm ready to receive some questions.
So hello everyone. Just a little ad before the Q&A session. Now we bring everyone back. So the registrations for the ASGCT annual meeting is already open. This year's annual meeting will take place in Boston from May 11th through the 15th. So you can already register on annualmeeting.asgct.org. You can do it today. So now we have a Q&A session with questions for all the speakers. We're bringing all the speakers back. Thank you for those wonderful presentations. Let me just access here the chat with our questions. So the first question for it's kind of for everyone. It's very interesting talks. Thank you for the overview. Following Maroon's landscape, how each one of you envision ways to integrate initiatives like the ones presented today. Is there any action to be taken to foster these interactions? And I think you can go in no particular order, but you can go like the order that you guys presented, maybe.
Yeah, just let me tell you that in fact we are collaborating for many, many years with people in Brazil. We are doing some working combination with CAR T cells right now. Experiments are running at Inca, Rio de Janeiro, with the group of Martin Bonamino and Dr. Veronica Lopez from our lab. So I think there is place for that. We did a lot of work with people from Chile for many, many years. So we need to find some ways to, and some subjects that would be common. And I learned just now that what Dr. Cooley just mentioned about the impact initiative in Chile. So I'm very glad to hear about that. And that's extremely interesting. And I would like to be in touch with that after this meeting. So that was very interesting, really. Thank you for that.
Dr. Curry, if you want to make any comment.
Sure. No. Thank you again for this session and the collaboration between ABT Cell and the American Society. I think I would like first to acknowledge the level of science that was presented by Osvaldo and Hilda, and this is a very cornerstone on how we can move to the clinic. As you know, there is the Pasteur quadrant where you have the basic or theoretical science with the Bohr quadrant, and then the Pasteur, and then the Edison quadrant. And I think that picture of very basic to translational to even market is even more merged with a new vision of let's combine all these knowledge to be able to move forward and have an impact. And this is a bit what the center is trying to do. Chile has since 2012 has taken the political strategy to look for long-term investment, because if you have a short investment in science of 2 or 3 years, you will have a gap, and then by the time you get another round of investment, then you lose your own people that you've trained for. So they created this center of excellence. They call them the Basel Center, and it actually give you a time frame of 10 years. You still have to look for investor, so private fund. You have to explain to them how this will work, and some of them are scared to collaborate with the academia. And then you show them some result that you are able to move from your scientific curiosity to translational to patient, and this is how you are able to provide the credential you want. And the government is giving those matching funds, so it's one-on-one. Sometimes it's 1-on-3, so it means like bring me 100,000, I'm giving you 300, etc., etc. And this is the kind of the long-term strategy. We're still way behind in terms of regulatory things in compared to Brazil, as I mentioned. But with the SWOT analysis I've showed, we call it in Spanish FODA, there are a lot of opportunity in terms of interest from people outside our region who are looking at South and Central America and are interested in having a fast access to the clinic, and interesting in and even situating their product in the region. And what I really like to emphasize on is the following: there are definitely opportunity. Definitely would like to collaborate with first. Sometimes it's good to see what's around next door versus going to the US, going to Europe, where probably our tendency is first to look outside, just because the reason it's easier to work when cells are manufactured in Brazil versus manufactured in the United States for logistics. And logistics is important when we're talking about CAR T, cryopreserving versus non-cryopreserving. And I think that initiative with webinars like this one, Guilherme, is very important to understand what are our capacities and how we can synergize. And yes, we can still work with North America, we can still work with Europe, but at the frame where looking at the necessity and what we need and what we cannot find in the region, and then reach out to the other. And then I think this is the mentality that we'll have to look at. And second point is disruptive. We have to be disruptive. If we are linear, then we will never gonna have CAR T. Many groups have been very creative on trying to do their own vectors in Brazil, on doing their own GMP facility, on cutting the cost. And I think this is a very interesting model that hopefully we can expand to the other country in the region.
Hilda, do you want to make any comment?
Yes, just a quick comment. Hi, everyone. Thank you for the question. Such an important question. I agree with the last comment that we need to know the environment in Latin America. We usually know everything about the US, Europe, but we know a little less about Latin America. And events like these are very important. In a Brazilian perspective, I think there's space for investments for that. And what we are lacking right now is investment from, we just have investment from the government. We be very privileged if we have investment for pharmaceutical companies to improve everything we have been done.
Great, thank you for the comment. We are actually reaching all the time limit that we had, but I would like to thank you, all the three speakers that we have today. Great presentations. I think this type of event like puts the seeds that we need to grow this area. And so I would like to thank you for attending the session of the ASGCT Around the World, the name of Technological Frontiers in South America in gene and cell therapy. And I think we accomplished that. I think that's exactly what we've been discussing here. Thank you, Dr. Osvaldo Podaiser. Thank you, Dr. Hilda. Thank you, Dr. Curry, for sharing your expertise and for the great discussion we just had. I think this is a good opportunity for us to start collaborations among Latin countries. So this event was recorded and will be available on demand on the ASGCT's website. I would also like to thank everyone at ASGCT's for the support, especially Renee Strong and Shelby House, which are here with us like in the backstage. So thank you, everyone, and we see you on the next event.